Hey, hi, everyone. Um, Last time we ended up talking about the function of enzymes, and we said that enzymes are a crucial part or a crucial type of molecule inside of our cells. Most of the time, enzymes are proteins, but we said enzymes function as molecular catalysts. Remember, what we were talking about was that the biological chemical reactions that take place inside of our cells that involve the breaking of bonds to allow the reformation of other bonds can either be in a process where you're taking complex molecules, maybe breaking them down into smaller molecules to release some energy, or the reverse of that would be taking smaller molecules and putting them together to make more complex molecules, and that would require you putting energy in. But we said those types of reactions typically would take a very long time to happen if you just let these molecules, bump into each other and hope that the right types of bonds breaking and bonds forming would happen on their own, right? Because these biological molecules that life evolved to use on planet Earth are what we call metastable. We said that biological molecules are pretty happy staying intact, indefinitely. Uh, and it's important to realize that they are metastable because life wouldn't work if you depended upon molecules that just fell apart by themselves all the time, because how could you control that, right? And likewise, if you had molecules that are really hard to break apart, that wouldn't work very well either, right? So metastable biological molecules are molecules that life evolved to make use of because they're sort of just right? They're stable, but they can be broken down, right? And they can be built up again. And we said that enzymes are the catalysts that make these reactions happen way faster than if you were just to sit around and wait for things to happen on their own. Remember, we said, for example, that in our cells, as our cells are doing their metabolic processes, they make lots of sort of waste products like peroxides, like H2O2, right? Which are just a natural part of different reactions happening. You're going to have hydrogens and oxygens coming together to form H2O2. But peroxide can be very damaging because those oxygens that are part of the peroxide, they're very greedy, right? They want to grab electrons from somewhere else. And so if peroxides were left to build up inside of your cells, they'd be bouncing around, just stealing electrons, messing up other biological molecules, right? A peroxide molecule might take several hours on its own to eventually break down into water, into H2O, right? But we cannot afford to just have our cells wait around for hours for peroxide to break down. We have enzymes like certain catalases that can come and really speed up the process of the peroxide breaking down. Remember, the example we looked at was a peroxide catalase enzyme that is the right shape to hang on to a couple of ions of iron, Fe, right? And it's also the right shape to bind to the peroxide. And by putting the iron and the peroxide together, the iron really helps to encourage the oxygen bonds to break, right? And it increases the rate of the peroxide dissociating into harmless water by something crazy like 300,000 times faster than if you just let the peroxide sit there by itself. So that's sort of where we left our discussion last time. And I emphasized to you that most enzymes are proteins, right? Because proteins, the stringing together of amino acids that are coded for by our genes, right? That's how you can get all kinds of wonderful, complex, and unique three-dimensional shapes once those polypeptide chains of amino acids fold up into the proper intended three-dimensional shape of the protein. You can have all kinds of little features, shapes, right? And that's why enzymes can work as small little machines that can bind something and catalyze or increase the rate of bonds breaking and bonds forming. So last time we didn't have time to look at this analogy, but I think this is a good analogy. So let's start with it this time. What is this, right? Yeah, obviously it's a stapler. So I want you to think about this, right? A stapler, let me hold it this way so it's just like the slide, right? So a stapler obviously is a simple machine, right? it helps us do a particular job a lot better than if we didn't have a stapler around. Right. And what job am I thinking about? Well, obviously a stapler, you say, well, it's designed to staple papers, right? So let's say I have a report here. So, I'm working on the MCB program, revamping the curriculum, and I have all kinds of little tables and things like that, right? And so the director of the School of Life Sciences, he says, Shen, I need you to put together this report, right? And I need it on my desk by the end of the week, right? So I think we all know that if you have a big report, you can have multiple pieces of paper, and I want to present this all together, right? I could just sort of stack these papers up, set them aside, and wait for the papers, the fibers of the paper, to naturally over time kind of come together. I mean, hey, that happens, right? I think we've all found a stack of old papers where they've kind of stuck together, right? And that might take what? I don't know, 10 years or something like that for the stack of paper to come together by itself. But with the help of a stapler, right, what would take maybe 10 years for the papers to stick together, I can do like in a fraction of a second by putting a staple in it, right? So I've catalyzed this reaction that I needed to take place because I could not afford to tell my boss to wait, yeah, give me 10 years and I'll get this report on your desk, right? So a stapler, let's think about this. It's a simple machine that dramatically catalyzed that reaction. Right? What makes up a stapler? There's a particular three-dimensional shape, and there is sort of the actual catalytic site, right? In this case, it's the particular site on the stapler where the staples are actually bent, right, and put into the paper. So remember, when we were discussing different types of membrane proteins that do things like transport solutes across a membrane in a direction that requires work, right? From low concentration solute on one side to high concentration solute on the other side, right? And we said that oftentimes those types of proteins, they break ATP in order to use the energy released by breaking the phosphate off of the ATP, right? Releasing that energy that's represented by that phosphate bond and using that energy to, let's say, make the protein change its shape so that it kind of forces the solute that it binds to go in the opposite direction, right? So that's work, right? I told you that, interestingly, all of those enzymes or those proteins that transport things, if they use ATP, we just think of them all as kind of generically ATP aces, right? So anytime you have a name of a protein that ends with an ACE, A-S-E, right, that suggests it's some type of enzyme. And so I said, isn't it kind of strange that even though they can transport different things, like one particular ATPase transports protons, another one can transport glucose molecules or other types of ions, I said, isn't it kind of boring that we all call them ATP aces instead of like a proton transportase or a glucose transportase, right? And I said, why? Because they all evolved to really do one thing, right? To break ATP, and that's really what they evolved to do. The secondary function of actually being able to transport something like protons or glucose, that is kind of like secondary, right? So let's think about this example here. If I say the stapler helped me staple these pieces of paper together, and I said, well, what's the substrate that this stapler works on? Right? Yeah, I'm obsessed with like tilting it the same way as in the slide here, right? I think many of us would be tempted to say, well, obviously the paper is the substrate, right? That's what this stapler works on, but that would be wrong, right? The actual substrate of the stapler are the staples themselves, right? The catalytic site of this stapler is designed to specifically do one thing, and that is act upon these staples, right, these generic staples here, right, and bend them in just the right way as at the same time it's pushing them. The fact that you have paper in the way is secondary. In other words, if I don't have any paper in there and I use my stapler, right? And it pops out a little bent stapler. Would I say, oh, the stapler didn't work? No, I would say the stapler still worked, right? So that makes you realize that the true function of the stapler is that it was designed to bend staples, not to put pieces of paper together. Right? Just like we said, remember that example when we were talking about ATPases, and I showed you the photo of that guy who has a jack and he's kind of raising up his truck or his car. And I said, think about it this way, right? You might say, yeah, that's a jack for raising a car, but in reality, you could put that jack under anything else, right? You could lift up a piece of furniture with it, and that jack would work just the same. Right? So that's why we say ATPases, like those membrane transport proteins, right? We just call them ATPases because that's what they do. They focus on breaking the ATP. The fact that they end up moving something else as a sort of secondary evolutionary change or ability, right, doesn't take away from the fact that they evolved just to break the ATP. So let's think about when you're talking about a simple machine, right, and you have to put some energy into it, whether you're talking about those membrane transporters or whether you're talking about our staple, right? Some energy has to be input into it in order for you actually to get the mechanical movement and the energy and the force to be able to do some work. So in the case of a stapler, that energy is, of course, my hand pushing on it. In the case of something like an ATPase enzyme, that energy comes from the breaking of chemical bonds, the breaking of a phosphate bond off of ATP. So a phosphate group from an ATP is often transferred, right, onto, for example, the enzyme, and that's what forces it to change its shape as the polypeptide chain that makes up this particular protein enzyme with all of its R groups from all of its amino acids. They're all trying to be comfortable, right? So there's a particular shape. You then stick a phosphate group onto them now, and they all kind of, there's like a ripple effect, right? The side chains kind of have to move around a bit, and as a result, the protein changes its shape. And just as long as the force of it changing its shape is stronger than, let's say, the force of the little atomic bond of the substrate that it's holding onto, then the enzyme kind of wins out, right? It might bend the bond of the substrate and cause a catalytic reaction. That bond of the substrate might break many, many times faster than if the substrate was just left alone, floating around without this enzyme kind of twisting it. So proteins that transfer phosphate groups, from an ATP, for example, onto another protein in order to activate another protein, those are what we call kinases. So kinases transfer phosphate groups to other targets in the process known as phosphorylation. Right? So you have protein A that maybe binds an ATP, right? And then you have protein B that comes along, and protein A twists the ATP, breaks off a phosphate group, and maybe moves that phosphate group onto protein B, changing its shape. We say protein B has now been phosphorylated by protein A, and protein A has acted as a kinase in order to do that. Okay? The opposite would be now you have protein B with a phosphate group stuck onto it, right? So maybe it's activated now, it's in the right shape to go ahead and do its function. Maybe you need to turn it off. So you have some other protein, some other enzyme come and remove the phosphate group off in a process of being dephosphorylated, right? And so that other enzyme that came around and took that phosphate off, we would call that a phosphatase that removes phosphate groups from targets. So let's look at this on a molecular scale. So this is a typical kinase domain of a kinase enzyme. So this is a protein, right? And maybe let's pretend this is a really big protein. So there's all kinds of extra, twirly secondary structures, tertiary structures of this protein here. But let's just zoom in and look at this particular region that's made up of these different parts of the polypeptide chain. All right, so in reality, let's say there are parts here that just go off the slide that we're not showing, right? Because in reality, this is a massive protein, but we're just zoomed in this particular region here. This particular region, the amino acid sequence makes it the right shape when it folds up to be able to hang on to ATP. So here's an ATP in here. So here's the ATP with its 3 phosphate groups. And at the same time, right, it's the right shape in here to bind some other substrate that it's going to do its kinase function on. Right, so the ATP itself, you might consider a substrate, and then you might have something else that's also a substrate that this enzyme, this kinase is going to work on. Right? So this would be the kinase's activation domain, right, where the action happens. So let's think about our stapler. All right, so our stapler's activation domain is this part here, this highly specialized part that's just the right shape to be able to act upon, right, this type of substrate, the staple, and you may or may not have another substrate at the same time participating, in this case, a stack of papers. So when you look at this and think about this in terms of what we see here compared to what we see here on the molecular scale, there are many parallels, right? If you think about, in order for a stapler to work as a stapler, what parts are sort of highly restricted in terms of what you're allowed to change or mess around with in terms of the shape, right? What parts are allowed to vary and still function normally as a stapler? So enzymes and their catalytic domains are highly conserved through evolution. In other words, if I want to design a new stapler because I don't like the shape of this one here, right? But I still want to be able to use these generic staples that I get from like, Office Depot or whatever, right? I'm kind of limited. So no matter what shape stapler you make, right, if you want to use the same sort of staples in them, then that staple activating site has to be the same, right? Evolutionarily, it's conserved in the evolution of staplers. Right? But the other parts of the stapler, you have more freedom, right, to change around their shape. And as a result, evolution would hang on to or conserve one part while allowing all kinds of experiments to happen through the evolution of life, through mutations, whatever, to come up with different shapes of the stapler body. And you end up with different staplers that work better for certain circumstances, right? So for example, this stapler here, my trusty swingline stapler, it's a nice heavy one, it's a quality one, right? That's why I can use it on my desk to staple like up to like maybe 30 pages together, and it works great. But I don't want to carry this around in my backpack. So really the one that I might want to carry around in my book bag is this smaller one, right? It can maybe staple 10 pieces of paper pretty well, right? Beyond that, it doesn't work as good, but it's way lighter. Okay, so think about this. In evolution, you would have a whole family of different related genes that are the kinases, right? Or I should say ATPases more specifically, right? And they all evolved because Mother Nature came up with, at some point in evolution, this catalytic site in this enzyme that can bind ATP and break the 3rd phosphate off. Hey, that's super useful, right? So that part is kept. And as through eons of evolution, as you have other proteins that might have resulted from those genes sort of being replicated and changing, different mutations would happen that would influence the other parts of the protein, enabling them to be able to interact with other components of the cell and be able to do this ATP breaking function in the context of different pathways. So let's look at this here. These are 3 different kinases, EGFR, HER2, and CDK7. Again, this is just zoomed in on their catalytic domains, right? And you can see they're very similar in structure because all of them kept this part the same, the ATP binding part and the breaking part. Other parts of these proteins would have varied just because of random mutations over eons of evolution. And as a result, they would actually interact with other proteins, right, that make their function different. So for example, EGFR, this kinase is involved in the growth of epithelial cells. HER2 has a very big role in the development of tumors. Right? And CDK7 is a regulator of cell cycle progression. all three of these, right, do their job by breaking ATP because they have the same catalytic domain, right? And then maybe putting that phosphate group onto something else, right? But because the rest of them is shaped differently outside of the catalytic domain, they're allowed to communicate with other proteins and be a part of very different cellular pathways: epithelial growth, tumor formation, cell cycle regulation. Okay. All right. So I want you to keep that in mind, right? That oftentimes many of these enzymes, right, have conserved domains because those are important functions that are used by the cell in different contexts. So let's shift gears a little bit and talk about metabolic pathways. I think by now you have an appreciation that life is a continuous chain of metabolic cellular reactions taking place, involving the building up and the tearing down of molecules, right? And that's why thinking about energy is so important. Energy that goes into things like enzymes that are piecing together other molecules to make complex molecules, right? And energy that's being released when you have things like enzymes breaking down complex molecules, and you have different molecules that are ready to sort of receive that energy that's being released. Recall that anabolic pathways involve the creation of complex molecules from simpler ones. We say that the flow of energy is endergonic, meaning you're putting energy into this system in order to build something up that's more complicated. So that's why the change in free energy, remember, free energy is just sort of a way of thinking, kind of abstract, about all the bonds that are involved here, right, that represent energy, potential energy that can be tapped into. Endergonic means you're putting more energy into making more of these bonds, energy going into the system. So it's a positive delta G. Catabolic pathways, on the other hand, are the breaking down of large molecules into smaller parts, right? So you're tapping into that energy represented by all the chemical bonds, and it's exergonic, meaning energy is being released from this system, right? And so it's a negative delta G, because then the products at the end, right, have lower free energy than the products that you start, or the reactants that you started with. So energy has been released. Right? And that energy, again, is either harnessed by the cell if you have other molecules at the right place at the right time to make use of that, to change their chemical bonds, or it's just lost in the environment, right, as heat. So in this example here, let's look at our friend glucose again. All right, let's start from the middle of this panel here. Here's glucose, C6H12O6. And we said that you can break the glucose, the carbon-carbon bonds down. And if you follow the blue arrow, that means you're going to have a negative delta G as you're releasing energy here. So it's an exergonic process. Maybe eventually you get it down to the most oxidized form that our cells use, the carbon dioxide and the H2O, at which point our cells have no more use for the carbon dioxide, right? So we release it from our body as a waste product. That's kind of like the end of what our cells care about in terms of how to be able to make use of carbon and oxygen bonds. Right? On the other hand, if you started with the CO2 and H2O, you could put in energy. Well, we cannot, right? But plants would take energy from the sun, and that would power the positive delta G of putting together carbon dioxide and water together into the glucose molecule again. Right? You don't have to stop there. If you take glucose molecules and then start putting them together, polymerizing them, that requires energy, right? Because two glucose molecules, they wouldn't want to necessarily link together. So you have to have an enzyme that comes, right, and actually facilitates that the bonds breaking and the bonds reforming, right? So that would also be energy put into it to get to the more reduced state, starch, right? Or in our bodies, glycogen. Right? And so this is the relationship here. Constantly, life is about going in one direction and going the other direction. And of course, the point is, inside of any of your living cells right now, there is a multitude of reactions taking place that are going one direction, a multitude of biochemical reactions taking place that are going in the opposite direction. So lots of anabolic processes, lots of catabolic processes all at once, So the sum of all of that, the changing energy, right, is metabolism, made up of anabolism and catabolism. All right. So this brings us then to how is energy actually distributed inside of our cells, right? I mean, it's one thing to talk about, yeah, okay, I get it, right? When you form a chemical bond between two atoms, that bond kind of represents some energy that could then be tapped into later if you broke that bond and allowed the electrons to form, let's say, a lower energy bond. Then you have that difference in energy that might be useful if you can harness it, right? So it's one thing to understand that, but how is it that energy is actually distributed and managed inside of our cells? So the key here is to think about fuel being converted into usable energy. Right? And I think one thing that students often get confused about is you know that glucose, right, is often referred to as a very important energy molecule in our cells. And you know there's this thing called ATP that's also considered a very important energy molecule. But how are they related? Right? Think about it this way. Let's say you start off with raw material. Right? Let's think about glucose as a raw fuel source. So glucose is the primary fuel for cells, but energy from glucose must be converted into a more convenient molecular form that the various energy requiring processes in the cell, meaning all the enzymes or whatever that's going to use this energy, are designed and expecting to use. Let's think about fossil fuels as an example, right? So you have a whole bunch of coal, for example, and coal is a raw fuel source, right? So the coal has to be processed by, in this case, this electrical plant, right, that burns the coal to release the energy from the coal and convert it into electricity, right? And then the electricity can be distributed, out of the city into your neighborhood, down the street into your home, right? And into your outlet. And then it's in the form of electricity that your devices, like your phones, expect, right? So I don't think you'd be confused at the idea that that electrical energy originally started off as the potential energy held in the coal. But the important thing to keep in mind here is if your phone is down to like one red bar and it's like 4%, and you're like, oh man, my phone is dying, right? If you had a friend who came up with a handful of coal and said, oh, here you go, charge up your phone, you can't do anything with that, right? You can't do anything with that because that's not the form of energy that your phone expects. Likewise, glucose is the raw fuel that's distributed through our body, through our tissues, and that's what our individual cells take up, right? They draw it in from the glucose that's circulating around in your blood, right? And then the cell has to convert that glucose into biochemical energy, right? The equivalent of the electricity that's flowing out of your electrical outlets on the wall. And that's what ATP is. So hopefully you realize now, glucose is very important. You can't talk about energy without talking about glucose because that's the raw fuel, but ATP is just as important because that's the form that that energy in the glucose gets converted into, so it can be distributed throughout your cell, and all of the biochemical pathways are designed, right, to use ATP, just like your iPhone is designed to use electricity from your wall. So the structure of ATP, we've talked a lot about ATP, but let's really take a closer look at the structure here. All right, so ATP is adenosine triphosphate, right? So it's an adenosine, so this is a nucleotide, with an adenine as the nitrogenous base, ribose sugar. If you just had one phosphate linked to this, then you could just stop there, right? Because this would be AMP, adenosine monophosphate, and this would be the building block of, let's say, an RNA molecule, right? But again, I always emphasize this, Mother Nature, right? Mother Nature, eons ago, billions of years ago, once things started being useful inside of early cells, they got used for different things. So in this case, if you add a second or a third phosphate group onto it, you go from AMP to ADP, diphosphate, to ATP, triphosphate, and this now becomes no longer useful really as a building block of RNA. This becomes the actual chemical form of energy that is derived from the glucose, right? So in other words, hopefully you can already figure this out, right? When the cell takes glucose in as the raw fuel source, as the coal being shipped to the electrical factory, right, or the power plant, right, the cell is going to process the glucose to convert it into biochemical energy, and you're going to be generating ATP as a result. So you're going to be putting phosphates onto these nucleotides to make ATP molecules, right? So ATP is the primary energy form, and it breaks down into ADP and inorganic phosphate, right? That third phosphate comes off. So here's this reaction. The ATP starts off with 4 negative charges on it, right? Here's 1, here's 2, here's 3, there's 4, right? And this third phosphate group that's stuck on there, it really does not want to be there, right? Just think about this. There's all these negative charges, so the negative charges are kind of like repelling each other. The phosphates themselves are very bulky, right? So there's a lot of like steric kind of hindrance as these molecules are being bunched up together. The first 2 phosphates are really kind of repelling the third phosphate, but the third phosphate is hanging on by this phosphoanhydrous bond, right? That's linking it and keeping it together. This is a very good example. This molecule is metastable, right? It'll stay as ATP, perhaps indefinitely, but it's pretty easy to break that third phosphate off because it doesn't want to be there in the first place, right? But you got to put some energy into it in order to break it off. Right? So this ATP here, it'll react with H2O, and it'll break down into ADP, right, the inorganic phosphate, and you'll have some protons left over. And in the process of doing so, right, you release 7.3 kcals per mole of energy. Likewise, so that's why it's going negative in this direction from left to right. Likewise, if you took ADP and you had some inorganic phosphate that you wanted to add on, that would require energy to do that, right? So that would be, it would take 7.3 kcals per mole to put together an ATP. Where does that 7.3 kcals per mole come from inside of our cells? Ooh, hopefully you figured it out. It's going to come from the processing of glucose, right? Breaking the carbon-carbon bonds of glucose and channeling some of that energy here into making ATPs that can then be distributed throughout the cell. So ATP is how energy is distributed throughout our cell. So think about it this way. Glucose is the raw material. It comes into the cell. It's going to get oxidized. It's just like the coal here being oxidized, burnt, right? You're going to have some leftover waste products, carbon dioxide and water. But in the process, you've taken ADP and free-floating inorganic phosphate, and you've taken the energy that's being released by breaking these carbon-carbon bonds in the glucose, and you're able to make molecules of ATP. And that is what's now going to be distributed inside of your cell, and all of these different enzymes and other proteins that expect to use ATP can get it, just like all the wires heading out from the power plant that's now carrying electricity, right, all throughout the city and into your home. And so that's the form of energy, ATP, that your cells will use to do all those different types of work that we discussed, right? Remember, there are 5 main types of work that are done by utilizing cellular energy in the form of ATP. Heat was sort of the 6th sort of way that biochemical reactions are useful for our cells. But we said that heat itself is just a byproduct of biochemical reactions taking place, right? And so we sort of think of it as not really being the intentional sort of function of breaking ATP down. It just becomes a natural byproduct that is very important for us. Right? So if we think about ATP as an individual unit of energy, you know, when you think about the electricity flowing from your wall into your iPhone through the little USB charger or whatever, right? It's hard to think about that as units of energy, right? But I mean, obviously there are certain units of how much energy you're using, because that's how your power bill every month or so is calculated, right? Like how many kilowatts of energy that you used. But when you're talking about biochemical energy in the form of ATP, that's easier to imagine one discrete unit, right? One discrete molecule of ATP is 7.3 kcals per mole of energy, right? So if you have a mole of ATP molecules, you get 7.3 kcals out of that mole, right? So we can think about it as currency, because individual units of something that have a value, you know, we're used to thinking about money, right? So not surprisingly, the different types of bonds that need to be broken and made as part of all the different biochemical processes that are taking place in your cell, some of those bonds, right, are much higher energy requirements compared to other bonds that are much lower energy requirements, right? And that all has to do with the nature of what molecules have these bonds that you're trying to build or break or whatever. So that's one way of thinking about ATP as currency. What can that one molecule of ATP buy you inside of the cell in terms of powering the creation of different types of bonds? Let's think about it this way. In this table here, you do not have to memorize this table, okay? But what they've done is they've listed a number of different molecular sort of, different molecules that are involved in different energy sort of pathways inside of our cell. And each one of these represents something like the opportunity for a phosphate to be broken off or moved around from this molecule. Right? Because the phosphate is really sort of, you know, the bonds that are being broken and reformed based upon spending a molecule of ATP. You're going to use that third phosphate to form different bonds with things. Some of these bonds involving the phosphate, right, are really high energy. So for example, this pyruvate, right, its bond that requires having a pyruvate molecule and adding a phosphate bond to it, that phosphate bond with this pyruvate, it represents 14.8 kcals of mole per energy. Right? So that means if you break that bond, you release 14.8 kcals, but the opposite would be it's going to cost you 14.8 kcals in order to make this bond in the first place. Then you can see they've ranked this in order of these different examples of what does it cost in order to get a phosphate to actually bind here, and likewise, right, how much energy can you get out of it, because it's the same thing, right? You can see that ATP they've put right in the middle here for convenience sake. So one way of thinking about it is this 7.3 kcals per mole of energy that you get from the ATP, let's pretend that's $1 US, right? Because we're all used to dealing with $1 US. Just like with converting currency, if you wanted to convert your US dollars to something like British pounds, right? Right now, the exchange rate is not very favorable. I think it's like, you know, whatever it is, 1.7 or something US dollars to get you 1 British pound. That means if you only have $1, you can't afford to get 1 British pound, right? You'd have to invest at least a couple dollars just to get 1 British pound with a little left over in change. So that means 1 ATP, likewise, right? If you break 1 ATP and get 7.3 kcals out of the energy, 1 ATP alone is not enough to go up on this chart here. However, right, there are many types of bonds that are lower energy than the ATP. So if you go down, you can go all the way down, right, to let's say something like glycerol phosphate, right? And so the glycerol, to be able to add or take off a phosphate group from that, only requires 2.2 kcals per mole. So that's like, if instead of worrying about trying to get a British pound out of your dollar, let's say you wanted to get some yen out of it, right? And the yen, right, has much lower value than $1 US, so you can get lots of yen just out of $1 US. So likewise, right, inside the cell, as it's going to be breaking ATPs, different enzymes using the ATP, right, in some cases, All it requires is a few ATP to be able to power the creation of certain bonds that are much lower energy. But on the other hand, it's going to require many ATPs to build up more complex bonds. Right. And again, think about it this way. These molecules, if they're all linked together in one pathway, right, this really represents the gradual release of energy as you start from the top down to the bottom, right? Going from sort of more reduced states to more oxidized states along the way. And each one of these steps would be mediated by certain enzymes that are the right shape to take the two things that are participating, right, and encourage that bond to happen. All right. So biological oxidation reactions then, going from high energy molecules down to lower energy molecules, are what we call highly exergonic. Right? Exergonic, again, we used that term before, means there's going to be energy released from it. Exergonic is not the same thing as exothermic. Right? Exothermic means that energy is mainly being lost as heat. Right? So we said highly exothermic reactions are bad because that's not compatible with life, right? Because that actually can generate a lot of out of control heat, which is not really compatible with our cells being able to function properly, right? But they can still be exergonic reactions just as long as stepwise, as you go down and have different enzymes taking care of each step, right? The energy that's being released is being harnessed, so it's not all lost as a crazy sort of release of heat, but instead stepwise little bits of energy being taken by different molecules. So from start to finish, it's still exergonic. You've released a lot of energy, but importantly, because it's designed that way to tap into that energy along the way, right? As you go from those different molecules down the table, for example, right? You don't get a lot of heat lost. That energy is hopefully being efficiently used. So carbohydrates, fats, and proteins are oxidized to release the energy from them. And so that means what? Electrons are being removed from them, right? That's what oxidation is. They are being oxidized as something else is being reduced, taking those electrons. So when you remove electrons, let's make this very clear. I mean, we're talking on an atomic scale. You're not going to have electrons that just pop off, right? And just sort of float around as electrons. Electrons are always accompanied by a proton, right? So really what we're talking about is when you remove electrons, you typically are removing a pair of electrons because remember, atoms, that's how they usually function when they're sharing electrons, right? It involves somehow a pair of electrons being moved around. And since those electrons are each kind of hanging on to a proton, you can think about it as, well, what's a proton with an electron going around it? That's basically a hydrogen, right? So we call this process dehydrogenation. Usually a pair of electrons and a pair of protons come off. Here's an example. Here's ethanol. Right? Here's ethanol, C2H5OH. So ethanol can be oxidized by our cells because ethanol by itself is poisonous. Right? It is a shape that we did not evolve to be able to tolerate having this in high levels inside of our cells because it's a little shape that gets in the way of other shapes functioning. Right? Remember, I told you, poisons are essentially just, they're bad for you because they're molecules that just happen to be a shape that your body can't deal with very well. Because unfortunately, these small molecules, right, are the right shape to maybe bind to other things and get in the way of important processes happening, therefore harming the normal functions of the cells. Right? So ethanol is like that as well. So ethanol has to be broken down through catalytic enzyme activity. Right? So let's say you oxidize the ethanol. So going from left to right, you oxidize it, right? So you've removed some electrons, and now that's why you now have the oxygen now double bonded left over, and you end up with acetaldehyde, CH3CHO, right? And you get a pair of electrons with their protons coming off of it, right? So that essentially is like hydrogen. So I want you to remember this. This pair of electrons and pair of protons that's taken off, they didn't just pop off of the ethanol, right? There had to have been some other molecule that came and actually took them off because that other molecule is the right shape, designed by evolution to be the right shape to bind to ethanol and oxidize it, right? And most likely that other molecule is some kind of a protein acting as an enzyme. Right? Those electrons need to be transferred to something else. It might be that enzyme that's come and done that, right? Taking the electrons off, and now it's holding on to those electrons itself. Right? So it's gained a pair of electrons and a pair of protons, right? So that molecule, that enzyme, would be reduced, right? Because it's gained these electrons. But if you think about this, great. So you have an enzyme that, let's say, takes electrons off of ethanol. So now the ethanol is on its way to becoming oxidized. First it becomes acetaldehyde, then maybe there's another enzyme that works on it, and another one. But this enzyme that ended up receiving the electrons, now what are you going to do with it? Right? Because maybe now that it's received the electrons, it cannot receive another pair of electrons. So what? Something else has to come and take those electrons off of it so that this can now function and work on another ethanol. Then what about this? Now this reduced molecule has the electrons, right? And so on and so forth. So that would not be very efficient if every enzyme that reduced something needs to have another enzyme that oxidizes it, right? Because then where does it stop? So that's not very efficient. So what's a better solution? A better solution is, what if the enzyme, when it moves the electrons off of the substrate, it doesn't actually accept them itself? Right? Remember, an enzyme can work on two substrates at the same time. What if there was some other small molecule that's also worked on at the same time as the enzyme, right? And the enzyme just transfers electrons from something like the ethanol onto this other little molecule, right? And this other molecule can just float away somewhere. Maybe it's a little bit more expendable, and then the enzyme is free to continue working. So enzymes often use another small molecule as the electron acceptor in these processes. And so that's what a coenzyme is. A coenzyme is a small molecule that helps enzymes function, oftentimes by acting as electron acceptors. So coenzymes are small molecules that bind to the enzyme at the same time as the other substrate that the enzyme is going to work on. And many vitamins are actually functioning as coenzymes. So let's look at this here. So here's a coenzyme, nicotinamide adenine dinucleotide, or NAD, NAD+. So here's the structure. So you can see it looks kind of weird, but it's two nucleotides, right? Can you see that? Here's one nucleotide down here with an adenine. Here's another nucleotide attached across the phosphate groups, but its group is this nicotinamide group. Right? So this is nicotinamide here. And the name nicotinamide, right, it's related to nicotine because this is the structure of nicotine over here. Right? But anyhow, this NAD plus group can easily take a couple of electrons with its protons and become reduced into NADH. Right? So this whole structure here is NAD plus. It can take a couple of electrons and become NADH. Thereby, the enzyme that's actually transferring these electrons around, the enzyme itself never worries about being burdened by having to hang on to the electrons. It's just given it to this coenzyme, and this coenzyme can float away somewhere. Right? So here's the structure of niacin, vitamin B3. Right? So this part up here would be what's up here. Right? So in this illustration, this is the nicotinamide because this is NAD, but niacin, or vitamin 3, would have this structure up here. And you can see vitamin B3 is important in your body because it acts as an electron acceptor. It acts as a coenzyme to help other enzymes do their function. So I think all of you at some point think about vitamins, right? You need to have a healthy diet with lots of vitamins, but did you ever actually think, right, what does a vitamin do? Vitamins are important because they help all of these enzymatic reactions happen inside of your cells. The more vitamins you have, the more efficient and the faster enzymes can work because they have places to dump their electrons that they're transferring. Right? Okay, this is the last slide. I know I'm kind of going over already, but I think you'll like this. Right? So let's talk about alcohol again. Right? Hey, who doesn't like talking about alcohol? Right? So again, here's ethanol. And so ethanol will be oxidized, right, by an enzyme known as alcohol dehydrogenase. So it's this big purple protein that binds to ethanol as its substrate, while at the same time binding to that NAD plus coenzyme. Right? And what it will do is this enzyme will catalyze the transfer of electrons off the ethanol to the NAD plus, thereby turning the NAD plus into NADH, right? Because it got protons along with the electrons, and the ethanol is no longer ethanol, it is acetaldehyde, right? So it becomes this. Then another enzyme comes along, aldehyde dehydrogenase, ALDH. And guess what? It acts on the acetaldehyde, right, with the help of NAD+, takes a couple of electrons off of that, converting it to acetate. And eventually, there are other enzymes that all take their turn, working on these different intermediate metabolic molecules from starting with the ethanol, all the way down to eventually breaking it down to carbon dioxide and water, right? So it can be breathed out, and it can be then peed out, right? So what this hopefully impresses upon you is that these different metabolic pathways all involve different enzymes, all taking their turn, working at the right time, right? And many of them are helped by these coenzymes. Around 80% of East Asians, people of East Asian descent, have a genetic variation in the alcohol dehydrogenase, this big purple enzyme up here, that converts alcohol faster into acetaldehyde, right? But unfortunately, around 50% of those same people, because of their racial ethnic background, also have a variation in their ALDH gene, right? That makes the ALDH enzyme that unfortunately slows down the conversion of acetaldehyde into acetate. So as a result, certain, people of Asian descent, myself included, right? When I have a few beers, right, I have a buildup of acetaldehyde in my body. And that acetaldehyde is poisonous, right? It's an intermediate that needs to be broken down. And it causes essentially an allergic type of reaction. You just think about, like, for example, formaldehyde, right? That's kind of nasty. You wouldn't want that in your blood. But acetaldehyde, similar type of thing. And you end up getting sort of a buzz, and you get allergy-like symptoms, right? You get the Asian flush, bloodshot eyes, runny nose, right? But the reality is, I think most people think, oh yeah, because Asians have a defect in their alcohol dehydrogenase. No, ironically, this is what I used to tell my friends in college. Ironically, right, I can break down the alcohol faster. I just get stuck on breaking down the acetaldehyde. So in reality, even though I look like a mess, right, I'm all bloodshot eyes and I'm bright red, the truth is I'm not even really feeling the effects of the alcohol as much. Right. So I remember in college, they'd be like, hey, look at Howie, right? He's totally wasted. I'm like, no, I'm not. I'm fine. Right. It's just that unfortunately, the bottleneck is the next enzyme, the acetaldehyde dehydrogenase. That's where my metabolic process gets stuck. Right. Okay. So sorry about going over, but I thought that that example would be very useful in getting you to think about, right, how enzymes need to work together in different pathways to be able to handle stepwise, right, all the molecules that go from complex molecules, for example, down to simpler molecules. And importantly, what each of one of those enzymes that takes its turn is coded for by its own gene, right? So different genetic backgrounds, different alleles of those genes could influence these pathways in different ways. And importantly, that these pathways oftentimes happen dependent upon the presence of these electron acceptor coenzyme molecules like NAD+ being turned into NADH, so that these enzymes don't have to worry about hanging on to the electrons that they're taking off of their substrates by themselves. All right. So let's go ahead and stop there.